So in the second of our tutorials on culturing microorganisms, I want to introduce you to the concept of a bacterial lawn. I'm going to show you how to prepare a bacterial lawn, but in reality, when you do this in your lab, it's quite likely that a technician is going to prepare the lawn for you, and you'll see why in a few moments' time. The other point I want to make here is that we're looking to measure effectiveness, okay? We're looking specifically to measure the effectiveness of antibiotics, or it could be antiseptics, okay? Antibiotics or antiseptics. I'm going to refer from here on in to antibiotics, but it will vary depending on the school that you're in, what you're using. And finally, in order to do that, we are going to measure what we call a zone of inhibition. And we're going to use the same formula we would use in maths to measure the area of a circle, which you probably know is pi r squared, and we'll come to that in a few moments' time. So this is kind of what we want to get to. Now, I'm not going to be as deliberate as I've been previously in exactly how we set up this experiment and these processes. If you do prepare the bacterial lawn in your school, you're very, very likely to be following a very, very specific protocol. So I won't bore you by going through that. What I would say in advance, though, is one of the main points I would make, other than the general equipment that we've got available here, is I'll just introduce you here to what we call the spreader. And it's this little implement here. We've just hand drawn it. And this spreader is an interesting thing because you've got to bear in mind, we'll just get rid of this a second, that your Petri dish is already going to have been prepared with the agar. The agar plate is going to be in the Petri dish. And what we're going to do here is we're going to use the spreader effectively to take something along the lines of 100 microliters of our bacterium, and we are going to use the spreader to spread it. There's no surprise there. And effectively what we're going to do is we're going to place it against we're going to open our Petri dish lid just slightly, and what we're going to do is we're going to hold the spreader in place, and once that spreader is kind of held in place there, what we effectively do is we move the Petri dish around until the 100 microliters of bacteria are spread. Just let me write that down. 100 microliters of bacteria are actually spread all across the agar, and at that point it's going to be allowed to rest at that point before we apply our antibiotic. Okay, so there's a couple of things there that I would just kind of encourage you to think about. I'm not going to go through this whole process. You can do that within your own practicals and your own studies. Okay, but that spreading technique prepares for us effectively our bacterial lawn. So what we want to think about really is that if we come down to our two examples here, our case 1 and our case 2, what effectively we have this kind of blue. Was that a blue or is that a green? I never good at telling the difference between blues and greens. We have this kind of blue. We have this blue here that's representing the bacteria that's been spread in the bacterial lawn. Okay, so just be aware of that. Now, the only other thing, and I'm using very much creative license here, we're actually viewing example number 1 from beneath. So we're actually, what we're going to do is we're going to take a wax pencil. We're going to take a wax pencil, and as you can see here, we are going to separate the plate into 3 approximately equal sections. Okay. The other thing is we're going to prepare a little circle in the center of each section, and finally we're going to put our initials. So for me it would be JS, and we're going to write on here, I'll put the date on for me right now. Today it's 15th of the 1st, 19. I wonder if that's going to be an interesting date because the Brexit vote is this afternoon. I don't know what's going to happen, but there you go, that's happening right now. And we're going to write on here that this is E. coli. Okay, now once we've done that, we're going to turn this over, and effectively what we're going to do is we are going to apply our our antibiotic discs. We're going to apply them onto these spaces. So we're going to take our antibiotic discs and we're going to apply them onto these spaces, onto these areas. So from above, although we wouldn't be able to see the lines because the agar would be in the way, we would have 3 antibiotic discs with 3 different antibiotics which would be placed onto the surface of the bacterial lawn. So just be reminded that blue, didn't get rid of my tool there, that blue actually represents the bacterial lawn. Okay, so just be reminded of that, the bacterial lawn. So, of course, what we're interested in is what impact is that antibiotic going to have on the bacterial lawn. Now, in case 2, we have a different scenario here. What we've got here is it's what we call an antibiotic disc ring. And you may well use one of these within your school. And the idea of this is that A, B, C, D, and so on, each of these rings, this is effectively a paper disc ring here, and each area A, B, C, D is soaked in a different antibiotic. So of course, once it's sat on top of our blue bacterial lawn, same on this side, we can look again at what is the zone of inhibition. Zone of inhibition. Okay, a nice way of remembering that, if you want to, is you can call that your Zoe. Okay, your Zoe, your zone of inhibition. And the idea of that is, to what degree does, for example, antibiotic B prevent growth and replication of the bacterial lawn, of the bacteria which has been spread with the spread of this blue bacteria. So, of course, if we get, and again, I'm using creative license to some degree, because, you know, if you were to get something like, you know, if you were to get sort of like a zone that the antibiotic didn't grow, something like that, for example, if you get a zone like that, we'd be able to say that B had been to some degree effective. Maybe on on E, you'd get a much larger area, much larger zone of inhibition, something like that, where we could say E has been more effective at preventing the binary fission, the replication of the bacterium. Okay, so here we've got some examples. If I just had those two zones, we could say A, C, D, F, G, and H were completely ineffective against this bacteria. Okay, so it gets kind of interesting from that perspective. So we're looking at this zone of inhibition as to what degree the antibiotics are effective at preventing that replication. Now, I was just reviewing my recording, and I realized one of the things I had not said to you was that the antibiotic actually kills the bacteria. I've been talking about it preventing growth. It kills the bacteria. Get that really clear and get it in your notes. So let's see if we can actually measure these zones of inhibition. In other words, actually measuring how effective these antibiotics have been, because they've been capable of preventing the bacterial replication or the binary fission of this bacterium. Okay, so let's call this example A, or antibiotic A. Let's make this B, and I'm actually not going to calculate C. It's just there because you're going to have 3 of these probably. So let's look at A. How are we actually going to calculate the effectiveness of this? Well, the key thing we want to do is we want to calculate the area of effectively this zone. Now we're going to treat it as a circle, okay? But what we want to do is we ultimately want to find the mean diameter. So we're going to measure that distance there, which of course is going to be the horizontal diameter. We're going to measure this distance here, which, you know, I guess we could call the vertical diameter, and then we're going to take the mean to get the overall diameter. So I'll just do it in a very simplistic way. Let me take our our ruler, and let me measure the length of this. Okay, so what have we got there? Effectively, what we've got there is we have a diameter of 20 millimeters. Okay, so we have got 20 millimeters across. Now, of course, what you guys are going to do is you're going to measure the vertical distance as well, and then you're going to add those two together and divide to get the mean. Okay, I'm going to assume that if I measured vertically, it would also be 20. So effectively, I've already got my mean of 20 millimeters. Now, effectively then, if we divide that by 2, that gives us our radius. Remember, radius would be this measurement here, or this measurement here. So the radius for me is going to equal 10 millimeters. Okay, now I'm sure we'll know from our math studies that if we want to calculate the area of a circle, we use the formula pi r squared. Okay, so again, thinking back to your mathematics, you probably know that you might use an acronym such as BIDMAS. You probably know that we have to do the indices or the exponents before we do a multiplication, because bear in mind, effectively, this is pi times r squared, what we're talking about here. So we have to do this bit first, because in our order of operations, we always do our indices or exponents first, okay? So let's do this. So let's do pi times, in this case, 10 times 10. Okay, so what we've got there for is pi times 100 is going to give us our answer. Okay, so let's actually do that on the calculator. Let's get rid of this. I was doing a little practice earlier. So we have got pi multiplied by 100 equals, and we'll round this to 314.2 to 314.2, and then we have to think about our units. Well, we are measuring in millimeters squared. So we're measuring millimeters and it's an area, so it has to be squared. So this zone of inhibition here is 314.2 millimeters squared. And we can judge that really to be the effectiveness of that antibiotic and the conditions that we've established. Now, let's look at example B. Bring our ruler back in a second. I've made the numbers very, very simple here. You guys will undoubtedly have different numbers here. So here, we'll measure our diameter as 10. Okay, so we've got this distance here is 10. Let's make an assumption that this distance here is also 10. You guys will measure it for real. So of course your diameter is 10 millimeters. We're going to divide that by 2 to get, again, the radius. Look, this distance, the radius. So our radius is going to equal 5 millimeters. Okay, now again, how do we do this? We do pi r squared. Just remind you that what this actually means is pi times r squared. Just remind you that that's going to mean pi times 5, which of course we know is 25. So we want to do a calculation of pi times 25, and that's going to give us this zone here of inhibition, this area calculation. So let's do that. Let's do pi multiplied by 25 equals, and we get a reading here of 78.5. Okay, so 70. So let's go for 78.5 units are super important, millimeter squared. So this zone of inhibition is 78.5 millimeters squared. Of course, this one would be some scales smaller, but the point we're making, of course, is that zone of inhibition A is significantly more effective than zone of inhibition B, because we've been able to measure the area, the zone where binary fission has not taken place. In other words, the antibiotic has prevented that bacterial replication, and that is the challenge we lay down to you.